All right. Looks like we are live for the SFT Creation versus Evolution podcast. This is episode 2. I've got Andrew C., aka a rational empiricist, here with me. I do want to thank everybody in the audience for being here for our late night podcast show on the creation versus evolution topic. Andrew, good to have you here. I appreciate you being willing to engage in these important discussions. Andrew, how you doing today?
Yeah, I'm doing good. Thanks for having me on. I'm glad I could make it to this discussion and glad to see everyone, the chat, willing to come here and listen to both positions and I guess mine in particular, since I'm the
You're in the hot seat.
right, yeah.
You're the
I
evolutionist.
blanked out on the word there for a second.
Listen, you found it. We found it together.
Yeah.
And again, I appreciate you doing this. I appreciate all of the late night party animals in the chat. No matter what time we go live, we've got a solid audience to enjoy these important discussions. So if anybody is wondering, we have been hosting so many debates on all sorts of topics: theology, soteriology, creation, evolution, nature of God, so on and so forth, that for myself, when it comes to engaging in these dialogues with those of differing views, since I obviously moderate these debates as we host them. So what I'm doing is this podcast series. So a couple of weeks ago, I had evolutionist Ken Rock here, and the topic was specifically ancestry. If you've not yet seen that, it is linked in the description box. Ton of fun. We went over 2 hours. It was comprehensive and touched on a lot of good topics. So here's episode 2. I've got a rational empiricist here, my arch nemesis. So, that's kind of an inside thing we have going on, but it also makes it fun for the audience. So, there's no secret that I'm a creationist. You're an evolutionist.
Yeah.
And the topic specifically tonight is going to be endogenous retroviruses, which is perfect because Andrew's favorite book these days, the Endogenous Retrovirus Handbook, written written by me. This is good for review and just kind of engaging some of the criticisms and objections in a sophisticated way. So Andrew, before we get into it, let me just address these super stickers, super chats. Doki Doki Bible Club, the super sticker master, appreciate it, brother. And George Bond, $10 super chat from Australia, appreciate it. Doki Doki is back. Yeah, baby is right. So Andrew, you know what, before we kind of get right into the topic, why don't we, for the audience sake, tell us a little bit about yourself. Sure. You're no stranger. You've been here before for informal debates and things like that. But a little bit about yourself. What's going on?
Yeah, so my name is Andrew, aka go by a rational empiricist. Currently a community college student here in California. I enjoy studying different topics related to biology, geology, astronomy, whatever I can get my hands on, whatever subjects, books I can get. You should see my library. But basically, yeah, I'm here to take the position that ERVs are evidence for common ancestry, specifically universal common ancestry, the very most common ancestry between all primates. I guess we could talk about that position specifically. And yeah, I think that there's not really that much to me besides that.
So what you're saying, Andrew, is you're a jack of all trades: biology, genetics, astronomy, geology. You do it all. You do it all, which is why you're perfect for this debate community that we have going on. So, as a debater in formal or informal platforms and format, it's good to have a wide range of knowledge on multiple different topics. Okay, well, why don't we why don't we start it off this way? So, since obviously we have differing views on the topic of ancestry and specifically endogenous retroviruses as they apply to the ancestry debate, right? Do the existence of endogenous retroviruses and even the nested hierarchies that they fall out into in sequences and in mutations,
Right.
are they better explained by the separate ancestry model or the universal ancestry model? So what I want to do, Andrew, since you are my guest for tonight, and again, I thank you for that, let's hand it to you. No time limit, no rush. Just kind of go over your position for the audience sake. Tell us a little bit about ERVs if you'd like to, and why
Sure.
you believe they're best explained by evolution. Go ahead.
Yeah, so I guess from the common ancestry perspective, or I guess the more mainstream biology perspective paradigm, ERVs, aka endogenous retroviruses, are viral DNA elements that are inserted into the host genome in a ancestral species, and if that ancestral species happens to, or sorry, I should say ancestral individual within a population, and that ERV fixes within the population, depending on whether or not it makes it to fixation. And from there, if the population within of that species happens to split into different populations, the ERV is carried along with it, inherited, we would say. And basically, from there, we get this natural nested hierarchy of ERVs within some lineages and different ERVs in other lineages. Basically, from my position, the way we know that ERVs are, among other things, viral insertions, is the fact that the genes, I should say, complete ERVs contain include the POL gene, the GAG gene, going out of order here, but yeah, the GAG gene, POL gene, and ENV gene, and those are usually found in retroviruses. More than that, we also have the long terminal repeats, aka LTRs, at the opposite, I guess, ends of the gene section, basically, for lack of a better phrase there. And apart from that, beside those LTRs, we also have what are called target site duplications. So basically, these are markers of where integrase, one of the retroviral proteins, kind of snips the DNA in two, and that is where it kind of patches itself into the host genome, essentially. You get these two repeating DNA segments on either end. I guess that's kind of the background of ERVs, and the reason I think they support common ancestry, or strong support for common ancestry, is the nested hierarchy, for one. There are kind of nested hierarchies within nested hierarchies here. Basically, the ERVs have shared locations, what we'd call homologous loci, between different species. And those fall into that distribution falls into a nested hierarchy. And as you kind of mentioned earlier, SFT, the shared mutations within these loci also fall into a nested hierarchy. And that, among other things, is kind of the main evidence that supports common ancestry coming from ERVs. And I guess one other thing would be that these nested hierarchies happen to support or are consistent with the nested hierarchies derived from other molecular sources of evidence, say pseudogenes and other genomic regions. And I think that has to be mentioned also because they could have happened to be different, but they're not. So I guess I'll leave it there. That's what I find to be the strongest evidence, and we can go from there as the discussion progresses.
Appreciate that, Andrew. Before I kind of give my thoughts on it in its entirety. You mentioned nested hierarchies within a greater nested hierarchy. I find that interesting. And for the audience sake, it might be good to define some of these more technical terms. What do you mean exactly by, let's say, a nested hierarchy, but more specifically, hierarchies within a hierarchy?
Right. Yeah. So a nested hierarchy essentially just means groups within groups. So if we're looking at the pattern of traits between species, we can arrange them, or this would be so good if I had an image to share here, but make things easier, but I'll try my best here. Basically, we can say, okay, all mammals, for example, have hair, among other features. Within the larger clade grouping of, say, vertebrates, all vertebrates have bony vertebrae, among other things, a backbone, essentially. And within all animals, for example, have certain traits, including non-vertebrates, that vertebrates share with them. So you kind of have this organization where these distinguishing traits connect groupings within other larger groupings based on more distantly shared traits. The technical term for these traits is called synapomorphies. Shared unique traits, I like to often call them. But yeah, that's essentially what a nested hierarchy is: a group, or is a system of groups within groups, basically. And to the other question, what I mean by hierarchies within hierarchies is essentially we can have a sort of a hierarchy, nested hierarchy derived from one line of evidence, say the shared location of these ERVs depending on the species. We can also have a nested hierarchy derived from the shared mutations within those ERVs. And essentially what I mean by hierarchy among hierarchies is these different layers of evidence for the same feature essentially corroborate each other. And like I said earlier, they also corroborate other lines of evidence, say morphology and other sources of molecular data. So that's what I basically mean by hierarchies here.
Again,
probably informative.
No, that's good. That's a great explanation. And so essentially what you'd be saying, like if we were to, as everybody understands, kingdom, phylum, class, order, family, genus, species, we have these, as you put it, groups within groups patterns where humans can be grouped with, let's say, the great apes in the great ape family, right? Individual species like the human species, the chimpanzee species, where they can also be grouped together as primates, grouped as mammals, vertebrates, with, you know, what the evolutionists would say, or taxonomists, a human, right, being a human ape, and like a chimpanzee and a gorilla being non-human apes. Humans and the great apes share more in terms of morphology, genetics, anatomy, and physiology than, let's say, the old world monkeys, fish, dogs, all the way down the line. I guess in its most basic form, would you say, Andrew, that's an accurate representation?
Yeah, I think so. Like I said, it gets kind of a bit more nuanced by the fact that Linnaean taxonomy isn't as cut and dry as we'd like it to be nowadays. And the fact that I mentioned these shared derived traits is what they're called. There's another discussion to be had basically between using shared derived traits and using shared what we call ancestral or primitive traits. But yeah, that's a fair representation of what a nested hierarchy represents.
And those shared derived traits would be the shared derived synapomorphies that you're talking about, correct?
Yeah, synapomorphies. The shared ancestral traits would technically be called plesiomorphies.
Right, right.
And there are other morphies besides that, but those are the two main categories.
So you would say, because you were pointing out these multiple independent phylogenies, that can be derived based on the evidence for endogenous retroviruses, right? Not only the hierarchies that the ERV elements fall out into, but also the hierarchies that the mutations found in the properties of the ERV element, like the LTRs, for example, right? You're talking about the long terminal repeats. There's another independent phylogeny that you're saying is best explained by common descent. Is that correct,
Right.
Andrew?
Yeah. Basically, you can find you could have found these shared ERV distributions could have supported a hierarchy where chimps and humans were not as closely related as, say, chimps and gorillas. But obviously, that's not the case. We find that the shared locations and shared mutations point to this group of chimps and humans within a larger grouping of apes, within a larger grouping of primates, etc. So yeah, they corroborate the same general order of branching.
So you're saying hierarchies within the great apes, hierarchies within primates, hierarchies within mammals, hierarchies within vertebrates.
Right.
Okay. So that's where we get the groups within groups patterns.
Right. I should make a kind of note here that at a certain distant relationship, or as you go back on the evolutionary tree, eventually these ERVs have been mutated to such a point that the groupings kind of falls away. So this is why we don't find shared ERVs, or for the most part, we don't find shared ERVs between, say, all fish, for example, because there has been, according to the evolutionary model, there has just been so much time since their last common ancestor. This divergence has caused the ERVs to essentially wither away, or to basically become unrecognizable. So it's only useful for certain groupings, but it does, I guess you could say, compared to the creationist model, it would show that what would usually be called kinds have these shared ERVs in common. So I guess that's one point that could be made, that it ends at a certain point, but either way it shows that these kinds have some kind of thing going on, basically.
So would you then say, Andrew, that your taxonomically restricted endogenous retroviruses, let's focus on just humans, so your HERVs,
Right.
human endogenous retroviruses, would you say that we would expect, based on the common descent model, that those ERV sequences would be more so complete than what you would say is a shared ERV sequence between a more distant common ancestor, let's say humans and a mouse?
Yeah, generally speaking, we can find that for the LTR sequences, for example. According to the evolutionary model, the two LTR sequences on opposite ends of the ERV sequence start out identical and diverge, become more different from each other over time. So yeah, generally speaking, the more time has passed, the more mutations they accumulate, as well, many other regions of the genome for that matter.
Okay, so you're talking about the LTRs, the long terminal repeats, right?
Right.
Is that basically when they go in looking for an endogenous retrovirus and also shared ERV sequences, right? When it comes to the hallmark signs that they would be looking to or looking for to say, okay, here's either a complete ERV or here's a degenerated ERV, right? An incomplete ERV, just pieces of the ERV, I guess. Is it the LTRs that would be the main sign or hallmark of an ERV? Is that essentially what they're looking for in these genetic sequencing?
It's one thing, like you said, if it's a complete ERV, for example, you'd obviously be looking for the retroviral genes along with the LTR sequences. But as I mentioned earlier, you also have to, or you're generally looking for the flanking DNA sequences beyond the LTRs, kind of like if the integration is basically an event where a scar has occurred, you're looking for the regions of tissue around the scar that kind of tell you where that scar is located, basically. And in this case, some of those sequences would be these target site duplications, which are kind of, for the audience, are these little short stretches of repeating. I wish I had a picture for this. Basically, when you have this certain sequence, for example, and since the DNA is complementary, when the integrase kind of snips it and inserts the retroviral DNA into the host genome, it kind of cuts it unevenly so that the opposite strand has to be replicated. And since DNA is complementary, you get two repeated identical repeated strands basically on opposite ends of the LTRs. And those are target site duplications.
So, and you're saying that sometimes the signal is a little more difficult in terms of locating these ERV sequences, because over time, the sequences themselves and the properties associated with them, they become more and more degenerate or eroded due to mutations over time, right? Like those LTRs, the second the exogenous retrovirus infects in the right way and is passed on vertically, the LTRs, right, the ends of the ERV element, you're saying, are identical. They have to be identical at insertion, correct?
Right. Yeah. Basically, even if you can have mutations, for example, where the retroviral genes are kind of entirely deleted, but you end up with these solo LTRs, basically, or there might be double LTRs. I haven't really looked at that specifically, but I do know that solo LTRs are remarkably common in our genome, for example. So yeah, essentially you're right there.
Okay, so we've got these ERV sequences then, right, being passed on vertically for millions of years,
right,
being subject to mutation.
I like the way you're describing it vertically there. That's a good way to put it.
Appreciate it. Yeah, especially for the audience sake, when you think of an exogenous retrovirus, typically you think of HIV, the HIV virus, right? Well, the HIV exogenous retrovirus is not passed on vertically, right, to offspring via genetics, but it is passed on horizontally, right? The ERVs, though, pass on vertically. Right. So if the exogenous retrovirus infects in the right way in a germ cell and gets passed on to the next generation, every single cell now has that viral genetic material, and now it's passed on generation after generation, where, as you're pointing out, the LTRs are nearly identical, but as time goes on and these ERV sequences are being passed on generation after generation, they're being hit with mutations and therefore becoming more divergent. Actually, this is where they would actually look to a molecular clock in the mutations in the LTR sequences, and determine a time for insertion. So over time, the signal becomes a little weaker because no longer do you have LTR, GAG, POL, ENV, right? You might just have what's called, you said, a solo LTR.
Right. Yeah. That's a good summary. Yeah.
Okay. So here's the next question I would have then.
Sure.
When it comes to the shared ERV sequences, right? Typically, that's what we'll say. You know, the argument goes shared ERV sequences. But in light of everything that we're talking about here, Andrew, how many of these sequences are actually whole, right? Complete ERVs, LTR, GAG, POL, ENV, versus just solo LTRs due to mutations and deletions over time?
Right. Yeah. So this kind of gets into the, I guess, depending on the researcher you're talking to, you can get, if they're defining an ERV, basically, like you said, as a complete ERV with all the retroviral genes and the LTRs together, then you still have, I believe, at probably at least a few 10,000, which if you're looking at solo LTRs, or just these LTR retrotransposons in general, which is another kind of way of uh, terming these ERVs, because they have these LTR sequences and they're classified as retrotransposons, hence LTR retrotransposons. Since they make up roughly 8% of the human genome, uh, as far as I'm aware, you find more of these degenerate ERV fragments, I guess you could call them, than complete ERVs.
Okay, so we
So,
find
so I
more,
don't know the exact numbers here, but I think that's generally the case.
and that's completely fine. So we do find more shared pieces of ERV sequences rather than shared complete elements of the ERV, is what
Right.
you're saying.
Yeah, I
More
think so, yeah.
pieces. Okay. But nonetheless, thousands, if we consider the few that are shared that are complete, but then the thousands, would you say pieces, solo LTR, so on and so forth, shared between, let's stick with the primates, shared between chimpanzees, bonobos, orangutans, gorillas? Um, so then are you saying that we would share more complete and incomplete ERV sequences as humans and chimpanzees, let's say? So we share more with chimpanzees than we do with gorillas. And we share more with gorillas and chimpanzees than we do with the orangutans. Is that your position and what you're saying?
Right. Yeah, that's the nested hierarchy we're talking about earlier.
Right. Okay, so we have a hierarchy in the elements and also a hierarchy in the pieces.
Yeah, basically, like I said, it depends on how kind of fragmented they are, how recognizable they are. But um, generally, yeah, for these, I guess you could call them small-ish groups on the evolutionary tree of life. Generally, you do find a large number of ERV, complete ERVs and ERV fragments that are shared between them.
Now, because endogenous retroviruses, because from my understanding, we share them pretty much with all vertebrates. Like we find these sequences in nearly all vertebrate genomes. Um, so if we're just looking at vertebrates, then how consistent is this hierarchy? Once you leave the great ape family, let's say once you even leave, the primate group, okay, um, how consistent is this hierarchy, or does it start to break down?
Uh, I guess since the main information or the kind of the main arguments centered around ERVs have to do with primates in general, I'm not terribly sure on that one. Sorry.
No, that's okay. I'd be curious.
Yeah, I'd have to look into that myself, but at the moment, I just don't know.
Let's even stick within mammals.
Sure.
It's safe to say that we share more with the chimpanzee and the other great apes than we do with mice.
Yeah, basically, yeah.
Okay. Okay. So more with chimpanzees than we do with, um, as we would find in the mouse
Right.
genome.
And
Okay.
to phrase it kind of a different way real quick, or kind of adding on to it, basically, you could also find unique shared ERV insertions in, say, rats and mice that aren't found in humans and other primates. That suggests that rats and mice, of course, are more closely related to each other than they are to primates, and that again falls into the nested hierarchy of these shared ERV locations.
Now, when it comes to these multiple independent phylogenies, Andrew, do we have the hierarchies primarily in the RTR, the LTR properties of the ERV, where we get the insertion? The LTRs are identical, and as time goes on, they accumulate mutations. So you have one LTR on either end of the ERV element, right? So you're saying, this LTR, let's say on the right versus the left, might accumulate more mutations than the left. So the right LTR becomes more divergent than the left, or what would you say, like the percentage? Is that what you mean by divergence, Andrew?
Uh, I guess it depends how you're looking at it. By divergent, I think you're just comparing one to the other. So it could be that one just happens to accumulate more mutations than the other. I think, at least from my understanding, the more likely outcome is they both accumulate a fair amount of mutations over time, but they're different, unique mutations. So, say you have an A to T in one base pair region, where you have an entirely different substitution in a different region of the other LTR. So I think it could go either way, but yeah, generally the discontinuity you're looking for between them, I guess, could account for either case.
Okay, so by different mutations, you're saying it could be like a base substitution, it could be a deletion,
Right.
an insertion. It's not always the same exact kind of mutation. It's not always just a base substitution, you're saying, or it's not
Right.
always a deletion, it's a mix.
Yeah.
Okay. Okay. Now, what about, because again, we're talking about the structure of the ERV, right? LTR, LTR, GAG, POL, ENV genes, or properties of the ERV. So we have hierarchies in the LTRs. Do we have hierarchies in the GAG, POL, and ENV properties of the ERV sequence? Like, where is primarily the hierarchies found?
So are you asking basically, do the genes themselves start out identical and kind of diverge like the LTRs, or I don't know if I quite understand your question.
Yeah, like when we talk about shared mutations in the ERV sequences themselves that you're saying fall out into these nested hierarchical patterns, that's the distribution of them. We have multiple independent phylogenies where we have hierarchies in the LTRs, mutations in the LTRs. What about mutations in the other properties of the ERV element?
Right, yeah. That's why I think adding the kind of unique portion to it. So these are kind of shared unique mutations. So if you imagine when you're doing your phylogeny, basically, you're often looking for, rather than traits that could appear, because you often can have evolutionary reversals, say an A to a T, but then a T back to an A, just because there are lots of nucleotides in the DNA that can mutate back and forth. But generally, you're looking for kind of these mutations that are less likely to occur, in the sense that I think I'm going off topic here, but yeah, basically what you're looking for is not just shared mutations. You're looking for these mutations that are found in one, that are unique to that one, and in a different ERV from a different lineage, a different location, you also have the exact same shared unique mutation going on. So say you have I don't know, just hypothetical example, you have a small insertion of a few DNA letters into one complete ERV in one species at one location. You have the same insertion in a different species at a different location in the same ERV, and that shows, or the chances of that exact mutation happening in that particular ERV at that particular location of the genome in two very to various extent related species is highly unlikely, and that's the kind of the hierarchy going on here.
Okay, so you would say then, based on
I don't
not
know if I
only
phrased that in a very understandable way, but you can try and help me out here.
you're doing good answering these technical questions. So we're getting in the important details, and I appreciate the fact that you're knowledgeable on this. Makes it more fun, right? Makes
Yeah.
it more of an engaging and
Right.
sophisticated
Yeah. Of
discussion.
course. Yeah.
So you would then say, I imagine, that given the hierarchies in both the ERV sequences and the mutational discrepancies and the distribution they fall into,
Right.
even considering the fact that we're looking at different kinds of mutations, right?
Right.
So for these to occur independently, right, in different lineages rather than them being inherited from common ancestors, let's focus on just humans and chimpanzees.
Sure.
Would one of the main arguments be that of statistical, right? You would argue that the chance of, let's say, a human and a chimpanzee, or even a human, a chimpanzee, and a bonobo being infected by an exogenous retrovirus that is then passed on vertically and now is an endogenous retrovirus. It occurs from within, right? It originates from within. To find these in the exact same spots and to have occurred independently is, as I've heard the evolutionists say, far less than 1 in 10 million. And when you consider multiple sequences that would have to be inserted independently in different lineages, then it becomes more like 1 in 100 million, 1 in 500 million, to the point where it's just very highly unlikely. Would that be an accurate representation?
Right. Yeah. And again, you can also extend this to the case of the shared individual mutations within these ERVs, for kind of the same reason. Like if you had a common ancestor, or say these two lineages, whatever lineages we're talking about, A and B, are separate ancestry. They're completely unrelated to one another. The chances of them having the same ERV insertion in the same location are vanishingly small. But the likelihood becomes even smaller, according to the argument, when they also happen to share the same exact mutations in their sequences. Because if they were not related, there's no specific reason beyond inheritance for them to share those exact mutations. So it's kind of, I guess you could say, the layers stacking on top of each other to build this argument is one way to put it.
So you're saying it's a multi-layered argument. It's
Right.
not just, okay, we have these shared viral genetic sequences between humans and chimpanzees and the primates and all vertebrates for the most part. It's more than just that. It's multi-layered. So I guess two questions.
Sure.
Because we discussed a lot there. When it comes to the shared mutations, when it comes to the hierarchies that they fall out into in both the ERV elements and the mutations themselves, and not to put you on the spot here, but obviously I know you don't have every technical paper handy, and that's fine.
Right.
What would be the best studies, the best technical papers that have actually documented these hierarchies that we find in the ERV elements and then the ERV mutations?
Let's see.
It's fine if you don't, because I'd really love to know, how many studies, right, comprehensively document this, because I also wonder behind the assumptions of mutations, because Andrew, you and I have discussed at great length the assumptions behind the universal ancestry and separate ancestry models, right? Where the universal ancestry model explains all DNA differences, all DNA, quote unquote, discrepancies as being the result of mutations over time, whether it's a frame shift, an insertion, a base substitution that we were talking about earlier, a deletion. This is all explained by mutations in one way or another. When the creation model, the separate ancestry model, would say, no, mutations do happen, but they are the minority in terms of genetic sequences, genetic elements, and DNA differences, right? We, who hold to the separate ancestry model, we would suggest that the majority of DNA differences found within species, across species, and this goes for DNA elements as well, like ERVs elements, are the result of initial creation, or design diversity, or in technical terms, created nuclear heterozygosity, right? So I guess my question to you is, and I find this shared mutation argument that fall out in nested hierarchical patterns to be very interesting, because I find it curious that in light of the evolutionists assuming that all DNA differences, all discrepancies between species, and obviously within species too, taxonomically restricted, that are the result of mutations. So what assumptions would you say the evolutionists are, what assumptions are at play when determining the shared mutational discrepancies that followed into hierarchies? How do you know that this, let's say, shared mutation or DNA difference is, a mutation, a base substitution,
Yeah.
an insertion, something like that? If you want to go into that a little bit.
Sure. Yeah, that's a fair point. Like, how do we know that these DNA differences are indeed from mutations and not from some other mechanism, whatever that mechanism may or may not be? Um, Basically, one thing I find, or one reason I think mutations are the dominant source of, I guess you could say, ultimate DNA variation, because we know that recombination scrambles or doesn't, uh, shuffles the DNA. You could say shuffles the variation around in a population, but ultimately, according to the evolutionary model, DNA differences come from mutations. And one reason why I think, a strong reason why I think mutations are the source of this DNA diversity. Oh, are you still there, Donnie?
Oh yeah. Yeah. Andrew, keep talking.
Oh, okay. Sorry,
Yeah,
I
you're
don't know
good.
if you
You're
left
good.
the
No
stream
worries.
for a sec.
No worries.
Yeah. So yeah, basically the main reason why we usually think that, or we assume that these mutations, or sorry, DNA differences are due to mutations, is the fact that from what we see of mutations that we have observed in the lab, for example, or just kind of a little background here, there are 4 DNA nucleotide types, basically A, C, T, G. But it turns out that these nucleotides, these base pairs, are not exactly the same chemically. So you have your purines, for example, and your pyrimidines, depending on whether there's kind of this 2-ring structure, this 1-ring structure. And it turns out that from mutations that we've readily observed in the laboratory, um, purines are more likely, not always, but much more likely to mutate into the other purine, and vice versa, than a pyrimidine, and a pyrimidine more likely to mutate into another pyrimidine, and vice versa, than into a purine. So this is, when you get into the phylogenetics, for example, just a side note, this is where in maximum likelihood analysis, you use these probabilistic models of sequence evolution where, for example, not all mutations are equally likely to occur. And that's kind of the one reason why I think mutations are the ultimate source of this DNA variation, is because when we look at, say, DNA differences that we haven't observed occurring in different species, we find that the occurrence of these DNA differences, compared to other sequences and other closely related species, um, which we can say, or hypothesize share a common ancestor, we find that they match the expectations of these probabilistic models. That purines mutating into other purines, pyrimidines into other pyrimidines more likely. And that isn't to say that a purine can't mutate into a pyrimidine at all. It just means that some DNA differences are vastly more common than others, and that's what we see, and why I think mutations are the ultimate source of these differences. So
Okay.
it's kind of long-winded there, but I want to go
Nope.
into
And that's
deeper.
fine. And that's important for the second half of our discussion. So,
Sure.
for the audience sake, what we're doing here is kind of laying down the details in terms of why endogenous retroviruses and shared endogenous retroviral sequences between organisms are important in the ancestry debate. Okay. So one more question, and then we're just going to do, as we do in this podcast series, a 2-minute break. I get a bathroom break from all the water that I drank, and then a refill. And then the second half, I am going to start, Andrew, and I'll give an alternative explanation. And then, for the separate ancestry model and explaining endogenous retroviruses, and then we'll engage that in detail. So my question is, when it comes to the shared mutational discrepancies that followed into hierarchies, okay, are these fixed? Like in humans, when you look at an ERV sequence and you see what you believe is a mutation, is it fixed within for the audience sake? Fixed just means to be stuck in place. Okay, so is this mutation, so called, is it stuck in place? Is it fixed?
Generally speaking, I believe so. So without going into the kind of the technical papers here, which I don't have on hand, basically, if it becomes common enough in the population, it becomes fixed. And you can kind of see this for these different species that supposedly have diverged so long ago, or relatively long ago compared to human timescales, that many of these DNA differences are presumably fixed, would be my guess.
Okay, so you have, let's say you have a common ancestor, right? You got the human and chimpanzee common ancestor between 6 and 10 million years ago. Okay, so they're hit with a bunch of exogenous retroviral infections, and then these insertions are passed on vertically. Now, eventually, you get a split, or you get the human chimpanzee split. So you get the human line going one way, you get the chimpanzee line going the other. With them comes the viral genetic material, right? The inserted DNA essentially is passed on. Once the split happens, now you start getting the possibility of independent insertions. So those would be taxonomically restricted, and independent mutations, right?
Right.
But the mutations that occurred in the ERV sequences prior to the split, right? Those are the ones that would be shared between humans and chimpanzees once they split and then diverge from each other. Is that correct, Andrew?
Right. Yeah. So you'll have these mutations are obviously shared between them according to this model, and you in each unique lineage you'll find their own unique mutations going along with them that aren't shared with the other lineage because they diverged since that time.
So you're saying then, for the most part, the ERV sequences that are shared between humans and chimpanzees and the mutations, since they were inherited from the past common ancestor, right? They would be fixed. You're going to find them for the most part in humans and then in chimpanzees, stuck in place overall.
Right. Yeah. For the most part. Yeah.
Okay. So I think that's important. We just hit the 50-minute mark. So, sure. I said one more question, but I do want to point this out in, especially again for the audience sake, and to make this podcast discussion as beneficial and edifying as possible. The most basic, I guess, mutate. I mean, you got different mutations, right? Deletions, base substitutions, insertions, frame shift mutations, insertions, so on and so forth. But the base substitution would be your most basic genetic mutation, right? And that simply involves the you were explaining it earlier, the swapping of one nucleotide for another nucleotide during DNA
you can
replication.
think of it, since these are kind of the smallest changes. One, they're the simplest to occur, and they're much less likely to disrupt anything because they're just changing one nucleotide for another, basically.
And how many okay, so how many of those are shared, would you say, on average, like a percentage? Or is that a big bulk of the mutation that's shared?
I don't quite know, to be honest, but I would assume so, given how common they are. So I do think it'd be a mix between the two. But yeah, I think since substitutions are generally more the most common type of, or for at least some regions of the genome, then yeah, I'd assume so for now, at least.
And I find that to be curious, since if you're just swapping out, right, one nucleotide for another, one DNA letter for another, I find it difficult then to truly determine if this is a mutation or if this is just a reflection of the functional nature of the ERV sequence. Because if it's just one letter for another, right, how do you know that that really did occur as a mutation? Especially if it happened in a distant common ancestor. And we'll touch on that in detail in the second half.
Sure.
But yeah, how confident would you be as an evolutionist to say, okay, this is most certainly a reflection of a mutation rather than, let's say, a created DNA letter?
Well, I think offhand, one reason would be the fact that you can have DNA differences, or minute ones, of course, because of the recent divergence. Like you said, you find unique mutations in one lineage in one ERV and not in the other. And I think that from what you're saying, if in my view, if they were designed for a specific purpose for that reason, that this wouldn't really be possible if they're highly specified here. Then, at least in my view, there shouldn't be these distinct mutations in either ERV and either lineage that kind of differentiate them. That show that there is variation possible, basically, in the sequence, even though it may be small or large, depending on the ERV.
Well, here's something that I find interesting.
Okay.
And this is the last comment I'll make before we go on. Just a 2-minute break here before part
Sure.
two. We're really going to get into the differentiating evidence and kind of engage it. These single base substitutions, or, in other words, these point mutations. Okay, from my understanding, a lot of rare diseases are due to single point mutations, right, Andrew?
Sure.
Sickle cell anemia, I believe, is due to a single point mutation. I mean,
Yep.
one single point mutation can kill a person. One single point mutation can lead to disease that, the person with the disease, it's harmful, but they can still survive. But the point is, and I'll make the point, I guess, by asking you a question, those point mutations that result in those rare diseases, would those be fixed in the population or unfixed?
Well, if they cause these diseases, then they'd most likely not be fixed because of the detrimental effects they have. In order for them to be fixed, they generally either have to be neutral due to genetic drift, or they have to be beneficial to be selected for.
Right. Good answer. So this is where I think the important aspect of this debate comes into play, in that we can determine, I think, safely what is actually the result of a mutation based on that which is fixed, that which is unfixed, right? I think we determined for the most part that, these ERV sequences that are shared are stuck in place. The mutations themselves that are shared are stuck in place, are fixed in the human population. Then we find fixed, so-called mutations in the chimpanzee population that are assumed to be inherited from a common ancestor in the distant past, right? But the question is, since they are fixed, maybe they are fixed for a reason. Maybe that is because they are a design difference. And that's where, in my book, I have a whole section that predicts that these shared mutations that form hierarchies, they may be pre-existing. They may be reflective of the functional requirements of the respective organism, right? Versus the unfixed mutations, which we can typically determine, like sickle cell anemia, or just mutations that have happened over, the last few hundred to few thousand years. They're not stuck in place, right? They're unfixed. So we can safely say, okay, this is, not a pre-existing difference. Essentially, this is something that has occurred after the fact. Have a quick final word on that. We're going to take a 2-minute break, and then we're really going to get into the meat and potatoes of this, Andrew. And I appreciate your answers here. Go ahead.
Yeah. Yeah, I guess that superficially would sound reasonable to me at the moment. I feel like I'd have some deeper responses here if it weren't so late back here. But yeah, I think I guess I'd agree with that for now at least. But I'll have to kind of look into it because I just to kind of make sure it's rock solid basically. So I'll give you that point for this discussion at least.
Okay, I appreciate it, Andrew. You did a great job answering a lot of these technical questions. We're going to do, we hit the hour mark. So typically these podcasts, we go 2 hours. We take a 2-minute break midway. So here we go, guys. Everybody in the audience, have some fun, have some side debates. Andrew, if you need a bathroom break or refill your water, now's the time. And 2 minutes. And, the battle continues. And the second half is always a little more wild than the first half. I'm just kidding, of course. But okay, here we go, guys. We'll see you in 2 minutes. All right, we are back and appreciate the audience for sticking in for the second half. And Andrew, no worries, we can even go half an hour. I know 2 hours is kind of long at this time. For the second half, I'll
do my best.
I'm even switching glasses.
I'll
so
try.
I got a new pair of glasses here, and they take a little while to get
nice.
used to. So, okay, here we go. I do got to say, shout out to Doki Doki Bible Club, the Super Sticker Master, putting my kids through college, brother. So I appreciate that. Okay, so I over an hour ago at this point now, Andrew, I appreciated your opening statement, and also the fact that you did a good job answering my questions to that. So what I'm going to do now is I'll just take equal time, about 5 minutes.
Sure.
We'll engage my points. A lot of this you've heard before, and that's why what we'll do is engage it. I'll present it for the audience sake. And then
slideshow ready.
There we go. I'll just go through a few random slides and also just reiterate some points from the first half. Okay.
Sure.
So, and this is why the topic of endogenous retroviruses as they apply to the question of ancestry is so important. Okay, guys, because as we demonstrated throughout the first hour of this discussion, if these really are the ancient remnants of past viral infections, okay, at least the pre-existing ones, that is, then it would be good evidence, reasonable evidence for common ancestry, because as we laid out, for all of these sequences to be inserted independently in different taxes, I guess you could say, different lineages, the chances of that, especially the mutations being the same mutations, is relatively low. Okay, and I can admit to that. So that's why I typically ask the question that is no stranger to Andrew. Are these really the ancient remnants of past viral infections, or are they created units of DNA function? At least the fixed ones. The ones that are stuck in place, I can confidently predict these are created units of DNA function. Therefore, I would predict function. And this also applies to the shared mutational discrepancies. Okay, if they are fixed, if they are stuck in place, then this may actually be reflective of the functional requirements of the respective organism. Okay, and what I mean by that is information flow and cell optimization would be expected to be slightly different in, let's say, humans and chimpanzees. There may even be secondary functions involved in these ERV sequences that are reflective of the so-called shared mutational discrepancies that may indeed just be created units, okay, design differences. And the way, let's say, a mouse would use its ERV sequence could be slightly different than the way a human uses their ERV sequences, and also, the chimpanzee, and so on and so forth. Okay, so all of this we touched on earlier. What is an endogenous retrovirus? Andrew also touched on the properties of the and the nature of the ERV sequences. Andrew, I appreciate you went into integrase. Right. We also have reverse transcriptase. Right. I find this interesting. Retroviruses, however, use a slower, stealthier approach. After entering the cell, the retrovirus uses an enzyme called reverse transcriptase to turn its RNA, because it has RNA as its genetic material, into DNA. It has to do that, of course, to insert and then hopefully be passed on vertically. Before making its way to the nucleus. Once in the nucleus, it inserts its DNA into the host genome, and then, of course, we get replication. And if it hits, as you can see here, most of the time when a virus integrates its genome with the host, a new hybrid genome dies when the cell and its descendants do. Sometimes, though, and this is what Andrew and I were talking about when it comes to the sequences that are passed vertically rather than horizontally, okay? Sometimes, however, a virus will infect a sperm or egg cell. If fertilization occurs, the offspring will have a copy of the viral genome in every single one of its cells. It can pass the hybrid genome on to its offspring, creating what scientists call a fully endogenous retrovirus, because it occurs from within, okay? A fancy term for a virus, as you can see here, that comes from within. We talked about HERVs, right? Taxonomically restricted endogenous retrovirus. The genomic organization and signatures of ERVs, what they look for, the LTRs, the GAG, POL, and ENV properties of it. And here's some important questions, okay? Why are there ERV sequences shared between the genomes of organisms? Well, we've discussed. It's either the common ancestry model would suggest that they're vestiges, and they can be utilized, or looked at as almost a historical record of evolution, common descent. Okay, or are they reflective of common design and design diversity, which has its own set of expectations and predictions? But if they really are created units of DNA function, even if we find functions, even if we find a lot of functions for these elements, okay, these created units of DNA function, as I would propose, there's still the greater question, and this is a good question. If viral-like sequences in the genomes of organisms are functional, why do they bear similarity to viral genetic material? We still have to explain why these ERV sequences, even if functional, still have these similarities, the LTRs, right? The GAG, the POL, the ENV. Why do they still have these similarities to exogenous retroviruses? And this is where I want to get to this slide. Okay, so for sake of time, I said I was only going to use 5 minutes, so I'll just go over this specific paper, but there are dozens of papers here. But this is what I find to be a more interesting and just fascinating function of these ERV sequences, in that they are antiviral protectors. Okay, they protect us from viral infections. This is 2021. So this is new research. And they still say in these papers, when you read them, that they're just in the beginning stages of understanding how endogenous retroviruses protect us from viral infections. There's still so much to learn, they'll say. They'll even say that, these are unexpected. And so they look forward to the future of discovery. What else are they doing? What is the, the nature of this ability, more of the details, essentially. So notice this excerpt from this paper. Long disregarded as junk DNA or genomic dark matter, endogenous retroviruses have turned out to represent important components of the antiviral immune system. Of course, for sake of time, I won't. I've got dozens of papers that talk about how they're functional in gene expression, gene regulation, embryological development. They're functional in, determining cell types, functional in tumor suppression, just a number of things, a couple that come to mind. Not only regulate cellular immune activation, but may even directly target invading viral pathogens. One focus will be on recent advances in the role of ERVs as regulators of antiviral gene expression. Okay, so here's where it discusses the tumor suppression. So the important point about their function in terms of being antiviral protectors is that it turns out that the way that the ERV is structured, their similarities to exogenous retroviruses are needed. Their function is actually predicated on their similarities to exogenous retroviruses, okay? Because they can perform something called viral mimicry as well, where the way that they're structured, that actually helps, are necessary for their function in fighting off retroviruses. And I wanted to go to one specific right here, for example. In what has been called viral mimicry, ERV expression can elicit host cell immune signaling via induction of viral defense pathways. It goes into a proposed explanation. Right here is where we talked about how 90% of ERVs don't even resemble true endogenous retroviruses, right? Solo LTRs that actually may just represent functional stretches of DNA. Notice this, but these similarities might be related to their function of fighting off viruses, right? They're similarities to exogenous retroviruses and are not necessarily due to some ancient viral invasion. Notice this, but it's also possible that what evolutionary biologists call endogenous retroviruses, remember the ERV sequences did not call themselves ERV sequences, okay? Scientists that assume they are the ancient remnants of past viral infections name them endogenous retroviruses. We're stuck with the name, so we'll stick with it here, although I think it's better too, in light of their many functional roles, to refer to them as created units of DNA function. Right here, the long terminal repeats that Andrew and I talked about may just simply represent long terminal repeats because they stretches of functional DNA can serve as binding sites for initiating or enhancing gene expression. There's one slide I want to get to, Andrew, and then I'm going to hand it back to you. And it is, let's see here. So again, the ERVs are integral and important components of the immune systems. And I got so many slides here that I may not be able to find it in time. But what I'll do is just go right back up to here. And for my last point, go over the mutations as well. So my overall prediction would be if we're looking at that which is fixed, okay, in the herbs themselves pre-existing, because we do know that one of the herbs jobs, one of their operational roles is to actually prevent the endogenization of exogenous retroviruses. That's interesting because remember, we also have DNA repair enzymes that stop mutations, right? That they fix mutations. And we have DNA breaks all the time. And if it wasn't for these highly intricate DNA repair enzymes, essentially we'd be extinct after a few generations. Of course, they work great. They work great, but they don't work perfectly. So mutations get through, mutations happen, many mutations in the somatic cell lines and in the germ cell lines where some are passed on as well, roughly 100 per person per generation. So the point I'm trying to make here is the pre-existing fixed herbs, they function to not only fight off exogenous retroviruses, but it looks like they also function to stop the endogenization process of them. Or even if an exogenous retrovirus becomes endogenous, is passed on, of course, the pre-existing ERVs, it looks like they can actually disrupt the endogenous retroviruses and pretty much kill them, so they don't induce harmful effects. I think we see this in the koalas with the KORV. Okay, so the point I'm trying to make is, although the pre-existing fixed herbs have that function, it doesn't mean that the function is performed perfectly, which means, yes, some exogenous retroviruses may make their way through, hit a germ cell, and through the fertilization process gets passed on. Okay, but here's the thing that I would say is those would be the unfixed ones. Those would be disease causing for the most part. And those should not have essential functional roles like in the embryo, and certainly not fighting off viruses. Okay. But it's the pre-existing, the fixed ones that I would predict are functional. Okay, functional to the point where they're essential, as in some of these papers that I have literally admit that without this ERV sequence, we would not exist. I like to exist. So here's the last thing on the non-random mutations and hotspots. Although I believe a lot of the DNA differences, a lot of the so-called mutations are reflective of design differences, okay, that may suggest the differences in the functional requirements of the respective organisms. But we do know that a lot of mutations are non-random. There are hotspots. And I would say that there are more hotspots than the evolutionists would say, because my position suggests and predicts that the majority of the genome is functional, where the evolutionists would predict that the majority of the genome is not functional. But if the majority of the genome is functional, that means these functional units would be expected to also be hit with a lot of the same types of mutations. Okay, if you actually Google, research what are the main areas most affected by corrosion or rust. Okay, and they'll talk about how it could be the paint of your car or the engine, and how there's on all modes of transportation that there's certain parts of the car that are more prone to damage, to corrosion. And even if it's an engine, we know the engine is important. Okay, and if we have all sorts of important and necessary genetic sequences and units in the genome, then we would also expect that a lot of them would be subject to the same kind of damage. So overall, the shared mutations, that's the last thing I'll say, Andrew, and then take as long as you want. The shared mutations, I think the answer to that is a combination of created differences, okay, and mutational hotspots. But I would predict there are more hotspots, many more hotspots than the evolutionists would predict. And that also comes down to whether or not the genome is mostly functional. And Andrew, that was a lot there. Go ahead, take your time. What are your thoughts on that? Anything you'd like to push back on? Anything you'd like a clarification over? The floor is yours.
Sure, yeah. So lots of diverse set of topics we can get into that unfortunately we may or may not get into all tonight.
We can do a part 2 and 3 as well. These are always fun with you, Andrew.
Yep. Sounds good. So I guess kind of pushing back on one of the main things you said, which might get us into another rabbit hole, but we can shift topics if need be from there. Basically, this conclusion that these ERB sequences are, like you said, not retroviral in origin. Because I think, like you said, if they are derived from exogenous retroviruses, then, in my opinion, that pretty much hits a significant nail against separate ancestry, which is why determining their origin is, like you said, so important for the argument. So if I could share my screen, I'd like to,
Absolutely.
for the audience's sake, show one thing I mentioned earlier, which I think pretty conclusively demonstrates that these are, in my opinion, retroviral in origin. Okay. Let's see. Is there any way I can make this any bigger? Does that make it any bigger on your end?
Yeah. When you zoom in, that definitely helps more for sure.
Okay, that's good. All right. Can you also see my mouse?
Yes. Yeah. I'll even make it full screen on my end too. There we go.
Okay,
That's
thanks.
better.
All right. So one of the things I was talking about earlier is the fundamental way integrase this specific enzyme that we know from retroviruses observed in the laboratory, how it inserts the DNA into a particular region of the genome. What it does is it basically cuts it at this kind of zigzag shape. It cuts it unevenly, basically. This is a clear delineation that integrase has been here kind of thing. When you find that, and the way we can track that basically is, okay, if say integrase has inserted this section of retroviral DNA into the host genome, what does this create? These gaps because of the zigzag shape that it's the uneven arrangement that's cut. And basically, since the DNA is complementary, you get these two identical strands that have been filled in. And these are called target site duplications. And along with the LTRs in the genes, they kind of show that the picture most consistent is, in fact, that at least for these complete ERVs, which are, as we talked about earlier, very still quite numerous in different lineages, these target site duplications, among other things, show that these ERVs are most likely viral in origin. So I kind of just want to show this graphic for the audience's sake to show what I was talking about earlier, make it a bit more digestible information. But yeah, that's the first thing I'd push back on is I do think these sequences are derived from retroviruses because of these target site duplications, among other things. So what are your general thoughts there?
Okay, so overall, you would say that these are, including the pre-existing fixed ones, the result of ancient viral infections, right, that have been passed on vertically due to not only their sequence similarity to exogenous retroviruses, right, LTR, GAG, POL, ENV, but also these target site duplications that you're talking about, right?
Right. So you can kind of, like I was saying earlier, you can kind of consider it if the ERV sequence itself was like a scar. These are kind of the regions of tissue around it that indicate a scar has been formed here, basically by some inflammation of some sort.
Okay.
In this case, the inflammation would be analogous to the integration for the audience.
So if these ERV sequences really do represent scars or genomic fossils, right, of evolutionary history, essentially, then these regions of tissue that we're looking at are evidence of that. Would be your position,
right? Basically, because we know that when we observe in the retroviruses today, HIV, for example, and all the other retroviruses, that the integrase it uses creates, like I said, this zigzag shape that leads to these target site duplications. And when we see these TSDs in the
I just want to clarify. You said
sure.
it's the integrase
Right, yeah,
that
it's the
result in these scar tissues, essentially.
right, yeah. Basically, the integrase itself is cutting and kind of inserting the retroviral DNA, and so it's kind of doing both at the same time.
Right, right. So the integrase, in doing its job, right, through cut and paste means,
Right,
or we know that a lot of these retro transposons can move around just through copy and paste or cut and paste, as you're saying.
right,
When this occurs, this is leaving every time almost a scar. Like if I were to fall on my elbow or something, and I were to get a scar, which I got quite a few scars on my body, it would be kind of silly to say that, these were pre-existing. I were born with them. It's probably better to say, no, that's probably due to an injury in the past. I fell
right,
on my elbow or something.
right. And I think yeah, that's a fair summary. I would say that it would be even more specific to say that, say, if we know from pre-existing conditions you land on your elbow in X way, you're going to get X scar, then that would show that okay, you probably landed on your elbow this way some time ago. And like we said, from these the LTR regions that seem to be consistent with this kind of the broader picture here, we can, like you said, take a molecular clock analysis and
Right.
kind of
Okay.
figure out the order basically of insertion.
Okay, so let's break this down a little bit.
Sure.
These are some really good points, Andrew, and clearly you've been studying this, and it makes it a lot of fun. So again,
Sure.
I appreciate that. So as I guess, where do we start? So as we've discussed, there are a couple of unique aspects of exogenous retroviruses, but we also find these in endogenous retroviruses. Are these specific enzymes, the reverse transcriptase and the integrase? Okay,
Right.
specifically focusing on the integrase here. Well,
Just quickly note, I will also note the ENV gene, just for science's sake, that these glycoproteins that for the retroviruses at least help them kind of gain access to the cell to begin with. So that's kind of
right.
the major proteins involved in overall. Just noting
Okay,
that. Yeah.
right. Okay, so in this process, then specifically of retroviral integration, right?
Right.
When the retrovirus makes its way into the host cell, right? And I got a whole section on this in my book too, where the RNA nucleic acid of the exogenous retrovirus, they're eventually released, and when reverse transcription takes place, right, which is pretty much just transcription in reverse. That's really all it means for the
Right.
audience sake. Okay, so the reverse transcriptase, it binds to the viral RNA to perform reverse transcription. But we also have the integrase, which helps to integrate the DNA of the virus, right, into the DNA of the host cell. And that's exactly why they would call it integrase in the first place. And then at the end of the day, we have
Insertase would kind of be weird sounding, to be honest.
integrase flows a little better. So now
Yeah.
we've got what's called a provirus, right, which
Right.
is a combination of host cell DNA and viral
Right. Yeah, that's a
DNA.
term we forgot to mention earlier, provirus, yeah.
Right. Now, so here's my question, and I'd like if you could reiterate it too, because this is such an important point.
Sure.
When we get these, these scars, okay, are these scars being left there? Okay, these signatures essentially for us to see when the either cut and paste occurs or the copy and paste.
So you're asking kind of, are these generally left for like say DNA transposons, which use the kind of cut and paste mechanism versus retrotransposons, which use a kind of more copy and paste mechanism. Are you asking, are they
Yeah.
found in both, basically?
Yeah, like because we know that, and I think we would all agree, the evolutionists and the creationists, that certain classes of retrotransposons, they can, one of their abilities is to move from one chromosomal location to another.
Right.
And what I find interesting, and I've got papers I could screen share, the ability of certain retrotransposons, not all of them can do them. It seems like they've lost their ability to mobilize, but many can. When they do move around the genome, they can not only turn genes on and off, but they can actually result in genetic variation that can be passed on. And what this tells me is there's a lot of latent genetic information in our genome, and because many of these retrotransposons, Andrew, have a gene promoter in them. Okay, so let's say we have a retrotransposon that's stuck in a certain spot within the genome. Okay, well, if they move, right, a gene that's turned on based on the location of this retrotransposon, if they move, that gene is now turned off. Right. So as jumping genes are popping in and out of the genome in different places, they can literally turn things on and off. And so my point is, when they do mobilize, when they do move around, okay, I would expect them to then leave these signatures, right? These scars. So the scars are being left from movement within the genome, but not from outside. Does that make, like, I would expect the same signatures from a retrotransposon that's moving in the genome, and if it came from outside. Like, how could we tell the difference? I guess my question would be, does that make sense, Andrew? Go ahead.
Yeah, that's a good question. So you're basically, just to clarify, you're asking these retrotransposons that are already in the genome, say, are when they're kind of copy and pasting themselves, do they kind of leave the same scars, these genetic scars, basically, as a exogenous retrovirus when it's first entering inserting itself into the genome?
Exactly. You got it. So when they move around in the genome from one chromosomal location to another, right, every time they do that, either through cut and paste or copy and paste, are they leaving the same scars that if an exogenous retrovirus was integrating from the outside? Would it be the same signatures, I guess, is the question.
Sure. Yeah. I as far as my understanding goes, I see no reason why it wouldn't generally be the case. I guess one thing I'd push back on, though, is that there are multiple types of retrotransposons, and obviously not all of them have these same genes, the integrase, for example, and LTRs for that matter.
Right. There's non-LTRs, you're saying,
yeah.
non-LTR retrotransposons, right?
Right. Yeah, exactly. So I guess my kind of counterpoint would be, it seems curious to me that these integration sites would also contain LTRs and these target site duplications if they're just from, say, any random kind of retrotransposon. Why do they happen to be these ERV-like sequences?
That's a good question. And I think it comes down to just how incredibly varied these ERVs, and other classes of retrotransposons are, and ALU sequences too, right? That are found in, I think, only primates.
Right.
Because even in a lot of these technical papers, like 2022, even, they're saying, like, wow, a lot of these functions are unexpected, and they admit, which is awesome, this is what science is about. They admit, that there's now so much more about these various classes of retrotransposons that we just don't understand. Like, as in, there's a strong level of excitement in terms of what more are we going to discover. I mean, we have some classes of retrotransposons. One specific that I like to point out that we find in the mouse genome, right? That if you deactivate it or snip it out, okay, the mouse, as it's developing, it stops developing and then it dies. Like it literally needs that piece of DNA, viral quote unquote, to develop. And then the same would be true for humans. That's that one specific class of retrotransposon. But then we got other classes of retrotransposons, like you're saying, where they can actually mobilize within the genome, right? They can move from one chromosomal location to another. And the presence or absence of some of these jumping genes can actually turn on and off various genes. And as I pointed out, some of these retrotransposons actually have a DNA promoter in them. And so one thing I would say too is their functional role in the innate immune system also relies on their similarities to exogenous retroviruses. And Andrew, that includes not only the LTRs, the GAG, the POL, the ENV, but the integrase and the reverse transcriptase. So that could be a possible answer too from our side, is everything about them in order to, and I used this analogy before, like if I was trying to, if an American spy was trying to infiltrate, actually, I'll use a different analogy. People that have watched like Goodfellas or The Departed or Godfather, all these cool mafia movies. Well, if you get somebody like an FBI agent who's trying to infiltrate the mafia, right? Or a mob, well, they got to look the part. They can't look like an FBI agent or else they're going to get detected and be killed, right? So they got to play the part. They got to look like a mob boss. They got to look like a criminal, essentially. And that's what we see as well with these functional roles in fighting off viral infections is they have to look the part. And they do. Thank God they do. And I would say that also suggests that they need the integrase, they need the reverse transcriptase. So yeah, there's a lot of variation in their functions. There's a lot of variation in what they do, why they're there. So it's not as black and white, I guess, Andrew, is a long answer short. It's not as black and white. So we'd have to look at the specific retrotransposon, and then we'd have to determine, what's the function, what's it doing there? Why does it? And I think once we recognize what the function is and why it's there, then we'll also know, okay, that's why it's structured the way it is. That's why it's an LTR retrotransposon versus a non-LTR, and vice versa. Go ahead, Andrew. Great questions, by the way.
Yeah, sure. So I guess this actually leads to some of the other questions I kind of want to push back on, which is regarding the LTRs and the shared locations, among other things. I'll start with the LTRs, basically. So you were talking about, for example, these DNA elements, when they're mobilizing, basically they're cutting and pasting, or copying and pasting more generally, into a different section of the genome. For LTR retrotransposons, specifically, these LTRs can, like you said, obviously serve as binding sites for proteins related to gene expression. I guess my question would be, if these were specifically created to do that, it seems like there'd be it seems curious to me, I guess I could say, that the designer would make it in such a way that we have these LTR regions related to gene expression, but we also have host genome promoters, like you said, among other things like enhancers and silencers that are also related to gene expression. And so it just seems odd to me, and I'd like to hear your thoughts on this. Why would the designer put these very different regions that kind of do the same thing but don't really need to be as different as they are in order to carry out their functions related to gene expression? So I'm essentially saying, if most eukaryotes, for example, already have these promoters, why do we need LTRs to be there also? Like, couldn't the designer have also made a different mechanism where he could have put promoters there already, or just a completely different way we haven't thought of, where it didn't have to involve these specific LTRs?
Those are good questions as well. I would say it just comes down to for one, the complexity, right? We're not looking at like a 2-bit code, zeros and ones. We're actually looking at a 4-letter code, ATCG, and a genome of essentially 3 billion letters, 6 billion in total, when 3 billion from mom, 3 billion from dad. So I don't think it's a stretch to believe that in the front loading of genetic diversity and functional DNA elements, there would be a variety of DNA elements that assist in a genome that is nested, consists of multiple overlapping codes and massive amounts of meta information. You'd have all sorts of functional stretches of DNA that are assisting in the gene expression pathway, determining cell types, gene regulation, so on and so forth, rather than just one specific genetic element that looks a specific way, and that is all that is used basically for gene expression. So I would just say, sure, like we find a variation. I'm not sure if that's answering your question. Are you saying why the LTRs that act as promoters for human genes, but also other types of genetic elements that also function as promoters for human genes? Like, why not just one? It seems redundant. Is that kind of your question? I want to make sure I'm answering it correctly.
Yeah. Essentially, you're basically saying that the designer made it for because of his creative diversity, essentially, right?
Well, especially because there's so much that is just going on right now in our bodies that we don't even know that that's happening. I mean, our bodies are regulating our heartbeat, our blood pressure, our brain activity, blood flow through the body. I mean, we're debating here, and yet we're thinking about what we're going to say next. And then we're thinking about what we're going to say next, as in, like, is that a good thing? Like, we're thinking about what we're thinking about.
Right.
Like, there's so much in our body that's going on in terms of regulating all the different systems, the nervous system, endocrine system, cardiovascular system. There's so much going on that I would also expect a variety of incredibly designed, unique, and complex DNA elements that help in that regulatory process. And one last thing I'll say is just the developmental process alone. Andrew, we have these various developmental windows that in 9 months we go from a single cell, a zygote, to a fully functioning, fully formed human being, a baby. And that developmental process requires so many DNA elements, many being the ERV sequences that are functioning right in development, determining cell types, determining what's going to go where and how it's going to grow, and making sure that there's not overgrowth, like making sure that you're not going to grow 10 fingers, on one hand instead of 5. So there's just so much going on to make sure that the human being develops and forms appropriately. And then a lot of those DNA elements are shut off. Their jobs, it's almost like a construction site. You have the construction workers that are actually building the building, let's say. Once they build it, they're done, right? They go on to another job, and then that building, whatever it may be, then you get workers in there and maintenance and stuff. So there's a lot in the genome that to an evolutionist looks redundant, but it's actually the fact that these elements, these redundant elements, have already done their job, and now they're either turned off, not expressed, or they're repurposed for a possible future job via environment, via stressor. But I'm saying a lot there, Andrew. So go ahead if there's some pushback or another question.
Sure. Yeah.
Feel free.
So kind of just to clarify, you are indeed saying though that the designer, if he exists, he could have this created potential of diversity in designing things in different ways depending on kind of the choices he's making, right?
I would think so. I mean, at my slide here, notice, the genome is a multi-dimensional operating system for an ultra complex biological computer with built-in error correcting and self-modification codes. I mean, we can go on and on about the multiple overlapping DNA and RNA codes. So yes, I would expect amazing variety in terms of the functional DNA elements that are actually designed and used for regulation, expression, so on and so forth.
Sure, yeah. So I think that gets into the last thing I wanted to ask, and we can go on a bit more about this before we wrap things up.
Sure.
Basically, if I guess my argument would be, if you're saying that the designer does have the potential to have this kind of created diversity in what he designs, as you said, with these LTRs versus other kind of promoter-like regions that we see in different organisms. And you also mentioned with, say, the mouse or the mice earlier. Basically, actually, you mentioned the let's see, development. I'm sure you're familiar with basically the placenta development due to how do you pronounce it? Syncytin. I'm probably mispronouncing it.
Syncytin, yeah.
Yeah,
Syncytin.
syncytin. Yeah, you know which one I'm talking about, basically.
Yeah.
The fact that we have, say, different syncytin ERV sequences in different locations in, say, the mouse versus the human, which shows that even though they perform the same function, they can be in different spots. But also, say, in ERVs that do have the same location or in homologous loci, for example, that it seems like you could explain that by variety, but it seems kind of contrived to me that, okay, he would design it in different places here, but wouldn't design it because of, like you said, his potential for created diversity. In why would he design it in the same location if he could also design it in different locations? It seems kind of inconsistent, if you ask me, based on what you've said so far.
Right. That's a very good question. Firstly, before I answer it in great detail, would you say that a lot of these, let's just say genetic markers, okay? Because there's a lot of different types of markers we could look at: pseudogenes, ERV sequences, okay, you name it. These highly conserved mitochondrial proteins as well, right? We can look to like the CO1 gene, or we can look to cytochrome C. You would say, correct me if I'm wrong, especially because one of your expertise, Andrew, is phylogeny and phylogenetic systematics. The reason why, let's say the CO1 gene, or let's say the cytochrome C, okay, one of these ATP synthase proteins, the reason why the human and the chimpanzee, let's say, although that protein has one function, right? One job. The reason why it's slightly different, though, there's a slight variation between the cytochrome C in humans and chimps. The reason is due to common ancestry, as in time since the split. For example, let's say you have a cytochrome C in the common ancestor, humans and chimpanzees split, humans go one way, chimpanzees go the other, then you'd expect some variation since the split. And therefore, there'd be more variation in, let's say, cytochrome C in humans and a mouse, because obviously humans and mice share a more distant common ancestor than humans and chimpanzees. So more differences in these highly conserved genetic markers between humans and mice than between humans and chimpanzees because of time since most recent last common ancestors. Is that an accurate representation, or just want to make sure?
Yeah, I think that's good so far.
Okay, so you're saying, why didn't God just make them all the same then? Why the variation that looks like common descent? Why not make them the same? Well, why sometimes in different
locations? I guess
locations?
just to clarify, to be specific here, I'm saying, why didn't he make them either all the same or all in different places? Like, why the kind of the combination between the two?
Right. And I think that's a good question, because you know what? That is what a combination of the two, that is what you'd expect based on common ancestry and divergence. But one paper here, this is what we're finding. Again, unexpected findings all the time. We're finding that a lot of functional stretches of DNA, including proteins. Like here's a paper, protein moonlighting, I find in nuclear DNA proteins to be rather fascinating. So notice this. A single protein with multiple functions might seem surprising, but there are actually many cases of proteins that moonlight or have more than one role in an organism.
Right.
Okay. Now here's the thing. We're just in the infancy of understanding the DNA language. Okay. So there's a couple things I have to respond to this, because this is a very technical argument you're using. Actually, this is probably one of the better lines of evidence for common descent. And it deserves a technical response. So here's the thing. Since we are in the beginning stages of understanding the DNA language, it's kind of like if I don't understand any Russian, I can't read it, I can't write it, okay? If I was hired to go to Russia to be a proofreader, okay, let's say they got a book, 300 pages, Donnie, I want you to proofread this. Tell me, what's a spelling mistake and what's not. I'm going to have trouble doing that because I don't fully understand the Russian language, and I'm going to be looking at it and I'm going to say, this looks like it's a spelling mistake, when in fact, maybe it's not. Maybe that's actually the way the word's spelled. And that's kind of how it is in the DNA language. It's very difficult right now for scientists to actually identify what is a mistake and what is not. What is a mutation and what is not? What is a base substitution? What is, an insertion, deletion, so on and so forth. That's how it, and that's why we as creationists, we say, no, we need to understand the fact that what we may be looking at is reflection of design diversity and the front loading of functional DNA elements. How this applies to your argument here is the reason why they don't all look the same and that they may be in different spots. Let's say they look the same in a human and a chimpanzee, or just slightly different, just a slight variation, but a lot different between humans and a mouse. And also it's in a different spot in the mouse than the human and the chimpanzee. Well, here's the thing though. If what I'm saying is true, okay, based on what we know about protein moonlighting, maybe other functional stretches of DNA have a primary function and a secondary function, with the secondary function being specific to the respective organism, having to do with information flow. So that means this, let's say an ERV sequence is in a different spot in the mouse, because although its primary function is embryological development, its secondary function may be different to the secondary function in humans and chimpanzees. And therefore, its primary function may be the same, but its secondary function is different, and so we find it in a different location. The question is, though, what is that secondary function? And that's where creationists can make testable predictions. I've made specific predictions on ERV sequences. Dr. Nathaniel Jensen made predictions on these highly conserved proteins that, maybe this or maybe that is the secondary function. That's a lot there. Go ahead, dissect that if you want. What are your thoughts overall? Do you think that's a good prediction? Go ahead. I accept all your criticisms, Andrew.
Yeah, sure. I guess the problem is there are multiple things I could touch on there, but I feel like if I do, then we're probably going to go over time.
The longest podcast of all podcasts.
Yeah. So I guess if it's okay with you, maybe I'll kind of make my final points. You can make your final points on kind of this general topic or maybe the specific one. And then we can either continue this at a part 2 maybe, or just kind of something like that.
If you don't mind this, Andrew, this for sure, we need an entire podcast for what I'm about to ask you. But I am curious because it does apply. Because I think we have about 3 or 4 answers to a lot of the questions you've asked, as in, why do they look so similar to exogenous retroviruses? Why are they designed with the integrase, the reverse transcriptase? Why do we see the scars, right? Well, the bigger question, okay, and a lot of evolutionists scoff at this model, but I actually haven't seen any really strong rebuttal. That's why I'm kind of curious, and I'd like to just get a maybe a generic response from you, and then we can touch on it later in a
Sure.
future podcast. Okay. The question is, what is the origin of retroviruses in general? Because retroviruses require a host to replicate. Okay. So the question is, did retroviruses come from outside the host or within? Right. So notice this slide. Accordingly, retroviruses could have originated from within the host genome instead of invading it from outside. This had to have been the case in any scenario, since viruses, this is what I want you to focus on, Andrew, since viruses are dependent upon host cells. This means that the cell's genome had to predate viruses. Viruses are incapable of surviving without a host cell. That is, the ERVs are more likely rogue products of the genomes of their host. So here's the thing. There's a strong case to be made that we have these variation-inducing genetic elements, okay, that you can call ERV sequences, and bad viruses actually originated from them, from within the host. And so the question is, if they are originating from within the host, and the host has these genetic sequences that are made of the GAG, the POL, the ENV, the LTRs, reverse transcriptase integrase, well, I feel like the evolutionists might have it backwards. The question is, why do the exogenous retroviruses look like the ERV sequences? Because if they actually came from the ERV sequences from within the genome, then it makes sense. That's why they look like them, because they came from them. They're rogue products. Now, a lot of evolutionists would scoff at it, and it's definitely not a prevailing model, but I think there's a lot of explanatory power in it. So I'd be curious as to your initial thoughts of that. And also, Andrew, how would you explain this paradox in terms of the origin of retroviruses to begin with? Did they... Right? Go ahead. What are your thoughts?
Yeah. So kind of unfortunately, you've hit on a major question, kind of conundrum in virology in general, which is, how did viruses at large originate if they're dependent on their host cell, whatever cell that may turn out to be? And I do think one of the, maybe not the exclusive hypothesis for their origin, but one of them, like you said, is that they originally came from the cell. Maybe it's genetic material of some sort. And I think that's one potential answer to your question, is maybe they didn't originate, or these retroviruses alone didn't originate from these sequences, but maybe at least a broad range of viruses in general, if that makes sense. So I think you're definitely onto something there. And I definitely will look into it, because that is a very interesting question, to be sure. But I think there's just so much research that still needs to be done, if that makes sense, regarding where specific viral lineages came from.
So you would say then that some viruses, there's many types of viruses, obviously. Some viruses, maybe the bulk of retroviruses, actually did originate from within the cell rather than outside, since they need a cell to replicate. That kind of makes sense, right?
Right.
Okay.
Yeah, I think that's definitely a plausible scenario, at least.
And here's the thing. This is exactly what we're saying with what's called the VIGE model, the variation-inducing genetic element model, that bad viruses that cause disease, that are harmful, HIV being one of them, are actually rogue products of that which they came from, and that which they came from was initially designed good. And that's why we see the ERV sequences do serve a lot of good. They definitely do serve a lot of good. So if they originated from the cell, then maybe the reason why they resemble, right, if they came from the cell, then the reason why they resemble the ERVs, then they have all these different components, is because, well, of course, that's where they came from. And obviously, there's a lot of study still in terms of that hypothesis.
And
but that too,
I have heard the VIGE hypothesis, basically. Unfortunately, like I said, I haven't had the time to look into it in as much depth as some of these other topics we've discussed, but I think that's definitely a motivation for I guess once I do that, we can continue from there.
Absolutely.
Yeah. So I think
that's
go ahead,
Andrew.
Motivation.
Yeah, I think you were saying, and I think that's great. I just stopped screen sharing. We're almost at the
Yeah.
2 hour mark. So Andrew, we made it. We made it.
Yep.
I didn't think Ken Rock and I would make it either. The difference between these podcasts is they're not formal. So there's no really long openings, long rebuttals. We're just
Right.
talking for 2
Yeah.
hours straight. Right. And that gets tiring, especially on a topic like this. It is so technical,
Right.
so important. And Andrew, you've been very sophisticated in your dialogue, been very respectful. So what I want to do is go ahead. Anything I said there, wrap up your thoughts, wrap up your points. I'll do the same, and then we'll wrap it up.
Yeah, so I think this is definitely an interesting question to look into. Are ERVs from common ancestry or separate ancestry? Obviously, I believe, as this discussion shows, that the most parsimonious explanation, when you account for all the data, is common ancestry. I will admit that there is always a small likelihood that they could have been designed, because science cannot rule out all explanations. But I do think that it can show that some are more likely to be true than others, in that some are more consistent internally, or more are simpler explanations. And I think, at least in my opinion, the data shows that common ancestry fits that category. But that's just my view, and definitely we'll probably have future discussions where we disagree with one another on that. But I appreciate the discussion we've had. The last one we've had kind of got a bit heated, and that's because there were multiple people in the room. But in this kind of format, I appreciate kind of the cordial back and forth we've had so far.
Absolutely. It's exactly why I'm doing this podcast. Sometimes in these open mics, it gets passionate. You got 10 different voices, and you got 10 different people wanting to make their point. So it's hard. And I really wanted to, this was important too, because it's something you can't do in an open mic, is spend an hour like we did understanding the argument, understanding what the problems are, understanding what we need to discuss. And that's why the shared mutations argument and the hierarchies that flow from them, it's such a deep topic that it's hard to give a quick answer. And that's why, we spend so much time discussing, the assumptions behind DNA differences. We discuss, fixed versus unfixed sequences and mutations. And what I'd like to say, though, for the audience sake, I just want to kind of wrap this up here. And
Sure.
Andrew, I understand that you're going to disagree with this, and that's completely fine.
Yeah, sure.
We can even do a greater discussion on it next time. But basically, okay, our model in its most simple form, all right, starts from Genesis. It's as simple as this. The Bible tells us that God created man in his image. Okay, so us as biblical creationists, at least, my approach is to say, okay, if this is true, if we really are made in the image of God, then there must be something about us that reflects the divine. As it applies to ancestry, universal or separate ancestry, okay, well, then maybe we can get a sense for how God designed the biological world based on how we design things. And this is the part that you love, Andrew. but and I say it because I think it, not everybody has to be an expert on this very technical topic to at least to understand this part. So here's the thing. I can look to the way we design things, at least currently. I think we're in the infancy of understanding God's design, and say, okay, and Andrew, this is where we're definitely going to have a greater debate. Okay, it so turns out that, there exists, in a sense, hierarchies in the design world, whether we look at computer code, the way systems are built, modes of transportation, things like that. We see human engineers build in homologous patterns. We see human engineers build interesting, what looks to be transitional forms. We see that definitely in the military, and we see that 100% with family vehicles. And I know as a family man too, like these crossover SUVs, it's like, man, what is that? It's hard to classify vehicles, and we find hard to classify animals. But here's the one thing I want to say. As it applies to, I'm setting up a possible answer to your question too, a more simple answer to why maybe God created an ERV sequence that functions in embryological development in humans, chimpanzees, and mice, but it turns out it's in a different location in mice. Well, you can actually look it up. There's vehicles where the engine has a function, a primary function for a vehicle, right? And I mean, without the engine, your vehicle is not going to function. It's worthless. But you can find an engine in certain vehicles that's built in the front, that's built in the back, and some, believe it or not, that are built in the middle. All right. So the question is, well, why? Why didn't human engineers just put the engine in the same spot in every single type of mode of transportation? You know, why is there variation? And the curious part is we're asking the same question about the biological world. So to me, this goes right back to my basic starting point. Maybe we can get a sense for how God designed based on the way we design. Okay. And I'll end it there. But what I typically like to do, Andrew, and this didn't work out this way, is whoever my guest is, I want them to have the last word. Okay. Because you were so willing to engage. So what I want to do is give you the last word. Okay. So if there's just a couple things you wanted to address, bam, we'll end it right after that. So Andrew, thanks for doing this. To the audience, thanks for tuning in. This has been a ton of fun. We're definitely going to do more of these. To any other evolutionists, but before you get the last word, Andrew, to any other evolutionists that want to do this as well, jump on the podcast. Let's pick a topic. You know, endogenous retroviruses seems to be the hot topic. So if you want that to be the topic, let's do it. I know me and Mark Drysdale are going to be doing one on that soon as well. Reach out. Let's set it up. This is a lot of fun, as I think you can see here with my discussion with Andrew. Andrew, go ahead. Final word, final thoughts, and then we are going to call it.
Yep. Kind of unfortunate that I missed a few seconds of your explanation there. My internet cut out for a short time there for a sec, but I did hear most of it. Yeah, obviously we are going to disagree on that. But I think it is good to examine the foundations of our ideas, especially in science, where sometimes the wrong assumptions can lead to the wrong conclusions. Obviously, in my view, sometimes when those assumptions hold, the conclusion probably holds too, but that's, again, another topic for another time. I guess I'll leave it at that. I've had fun with this discussion on ERV so far.
All right, Andrew, appreciate it. Make sure you send it to my boy Speed of Sound.
Right.
Would love to hear his thoughts.
Yeah, he's probably gonna have something to say about it when he sees this. So
no worries.
Hey,
I appreciate listening to the critics because it's how I improve as well, and it helps with my research and study. So, Andrew, appreciate you jumping into this podcast. It's not always easy, but we like to uphold and promote critical thinking. So we just want more people of differing views to feel comfortable in jumping into the octagon and engaging in these technical and important topics. Okay, so the SFT, Creation versus Evolution podcast, episode 2 is over. Share it around. Hit that subscribe button, reminder bell, notification bell, and God bless all. Stand for Truth is out.